The microbial world is massive, and we are exposed all the time to bacteria, viruses, fungi, and parasites. And most of those organisms are either commensal organisms that interact with humans harmlessly or environmental organisms that don't interact with humans at all. But there are a subset of these organisms that cause disease, and we call these pathogens. Our immune system is geared towards recognizing and eliminating pathogens, but not everything else. And so that means our immune system has to do two things. One, it has to distinguish between self and non-self so that it can protect us from infections or things like tumors, but not cause autoimmune diseases. And second, our immune system has to distinguish between dangerous and benign so that we make responses to pathogens, but not to harmless things like commensal organisms or food or things like that. Our immune system does this largely through white blood cells that originate in the bone marrow. These blood cells are derived from hematopoietic stem cells that give rise to common lymphoid progenitors and common myeloid progenitors that in turn give rise to lymphocytes, such as T cells, B cells, natural killer cells, and dendritic cells, as well as myeloid cells, such as neutrophils, eosinophils, basophils, mast cells, and monocytes. The neutrophils, eosinophils, basophils, and mast cells are collectively called granulocytes. And because of their strange nuclear formations and segmented nuclei, neutrophils, eosinophils, and basophils are sometimes called polymorphonuclear cells. Monocytes are present in circulation, but as they migrate into tissues, they differentiate into macrophages. So we do not find macrophages in circulation, but they are an important cell type of the innate immune system, whereas we find monocytes in circulation, but we don't find them in tissues because they convert to macrophages once they get into the tissue. Our immune system can basically be divided into two components, the innate immune system and the adaptive immune system. Our innate immune system can be considered to be composed of physical barriers like skin and mucosa and epithelium. Our innate and adaptive immune system also have cellular barriers. Many of the myeloid cells that are either phagocytes or granulocytes or natural killers I mentioned in the previous slide. And the cellular barriers of our adaptive immune system include things like B cells and T cells. One key difference between the innate and adaptive immune system is that the innate immune system response is immediate, whereas the adaptive immune system response takes a few days to get going. There are some important secreted molecules in the innate immune system, things like the complement cascade, which we'll learn more about later, pore-forming molecules like defensins, enzymes like lysozyme that break down microbial products, whereas the secreted molecules of the adaptive immune system include antibodies and cytokines, which we'll also learn more about in just a little bit. The receptors that recognize foreign antigens in the innate and adaptive immune system also differ. In the innate system, all of our receptors are DNA encoded, and they are broadly specific. But in the adaptive immune system, our antigen receptors come about through DNA recombination, meaning every T and B cell has its own unique receptor, and each of these receptors is finely specific for a very particular antigen. Lastly, our innate immune system does not form immunological memory, but our adaptive immune system does, and this forms the basis of immunity and is the rationale behind the development of vaccines. Let's get a little deeper into the cells of the innate immune response. As I mentioned, neutrophils and monocytes and macrophages are important cells to the innate immune response. The neutrophils are the most abundant and are both in circulation and in the bone marrow. And these cell types have antibacterial, fungal, and parasitic functions. Natural killer cells are much less abundant, but are important in the antiviral response. And lastly, eosinophils, basophils, and mast cells are important in the response to certain parasites, such as helminths, as well as important participants in inducing allergic responses. The cells of the adaptive immune system are highly specific and can respond to all types of pathogens. I've drawn a picture here of a skin that is compromised and allows for the entry of bacteria into the skin as kind of an example of how the innate immune system gets activated. When macrophages within the skin are exposed to foreign pathogens, they are induced to produce a subset of molecules called cytokines that induce the inflammatory response. Dendritic cells, on the other hand, migrate to lymph nodes, and there they are important in activating T cells. Lastly, there are soluble components like complement that are also activated by exposure to pathogens, especially bacteria, and can enhance the inflammatory responses in those tissues. We'll focus primarily on complement and inflammatory cytokines. Now, what is a cytokine? It's a small secreted protein that controls growth, function, and activation of immune cells that express the appropriate cytokine receptor. Cytokines can be present all the time, or in other words, they are constitutively expressed, or they can be induced under certain circumstances. And they communicate between cells of the immune system. Chemokines are a subset of cytokines that have a specialized function in inducing cell movement or chemotaxis. And this is the movement of cells from areas of low chemokine concentration to areas of high chemokine concentration within a given tissue. Now, within an inflamed tissue, inflammatory cytokines produced by macrophages have a variety of effects. They activate local capillary endothelial cells and induce vasodilation. When this happens, this allows rolling leukocytes within the capillaries to adhere firmly to the activated endothelial cells, squeeze between the tight junctions, and migrate into tissues. Once they are in those tissues, they can migrate along a chemokine gradient towards sites of inflammation where they can mediate important effector functions, including phagocytosis, or the ability to eat or consume pathogens, as well as secretion of toxic granules. Now, dendritic cells within tissues also are able to become activated through exposure to pathogens, but when they become activated, they carry their payload to draining lymph nodes, and they do that by migrating through lymphatic vesicles to the draining lymph node. Lymph nodes are connected by a series or highway of lymphatic vessels that transfer lymph from tissues into the draining lymph node. This lymph can carry dendritic cells that are presenting foreign antigens, but they can also carry pathogen products, as well as the pathogens themselves, into the draining lymph node. Once these products are in the draining lymph node, they can activate T cells and B cells. T cells are activated by dendritic cells that present pathogen products to two types of T cells. One is the CD4 positive T cell, and the other is the CD8 positive T cell. The CD4 positive T cell, when it becomes activated, becomes a T helper cell that secretes many important cytokines that mediate immune system activation, that activate innate cells, and that activate B cells. And these T helper cells have important antibacterial, viral, fungal, and parasitic functions. CD8 T cells, on the other hand, acquire the ability to kill infected cells and are primarily important in the antiviral response, although there are some exceptions. B cells, on the other hand, can be activated by soluble molecules or even pathogen surfaces. And when they get activated, they can differentiate into plasma cells that produce large quantities of antibodies. These antibodies have a variety of functions that we'll learn about in more detail another time, but this includes the ability to neutralize pathogens, or in other words, prevent them from infecting cells or colonizing. Antibodies can activate innate cells, and antibodies can activate the complement cascade. The types of antigens that are recognized by B cells and T cells also differ. B cells can recognize free-floating antigens, anything that's free-floating, including proteins, DNA, lipids, and polysaccharides. T cells only recognize small amino acid chains called peptides that are presented on the surface of an antigen-presenting cell, like a dendritic cell, within a complex of molecules called the major histocompatibility complex, or MHC. When the MHC is able to interact with the T cell, it induces their activation. In additional video sessions, we will discuss the key aspects of the innate and adaptive immune response that give rise to both broadly reactive inflammatory responses and highly specific responses mediated by antibodies and T cells.